Hi everyone, we're going to work through the digestive system in this lecture, and we'll start off with an overview of the anatomy of the digestive system. So the digestive system looks something like this, and there is basically one long tube about 30 feet long, which is called the alimentary canal, and that extends all the way from mouth down to anus. And along the way, there's going to be different extensions, different morphological features. We'll talk about those on our tour of the alimentary canal. But in general, it's all the same type of tube. There's very similar features all the way down in terms of the composition of the tube and how it's arranged. And then furthermore, above and beyond the alimentary canal, there are a couple of accessory organs of digestion. And those would be the liver, the gall bladder, and the pancreas, which are going to not be a part of that alimentary canal, but nevertheless their contents will be injected into the canal at some point. So they become accessory organs. All right, here's what the alimentary canal looks like. There's four main layers. There's the mucosa, the submucosa, the muscularis. This is also sometimes called the muscularis externa. And then there is the serosa. Okay. Now, a little bit about these different lamina. In the mucosa, there's an epithelial tissue that lines the inside of it. And there is then the lamina propria, which would be a connective tissue that is underneath it. Now, this is a looser connective tissue that allows things like glands and lymphatic vessels to pass through it. So it's not as dense with the tight cell junctions as you would see in the epithelium. By the way, the epithelial tissue can vary. Generally in the esophagus, it's a stratified squamous epithelium, and then once we get into the stomach and then the small intestine, it transitions into a simple columnar epithelium. Is the predominant epithelial tissue found all the way along. Okay, in the submucosa, there is a plexus, and what a plexus is, it's a network of nerves that branch, and one signal from the nervous system could come down and sprawl and branch and wrap all the way around the layer. And so it makes it easy for things like glands to secrete their contents, whether this be enzymes or mucus. One signal coming in from the nervous system could spread throughout the whole sheath and cause multiple glands to secrete their contents at once. So there's more uniform central control of that region. A lot of the glands that make their way through the lamina propria of the mucosa, they originate in the submucosa. And this is where also a lot of capillaries would be found, where blood is going to take up nutrients and things. Okay, wrapping around then the submucosa is the muscularis, and this actually has two components to it, the circular layer and the longitudinal layer. So the circular layer is sets of rings of muscle that wrap all the way around the tube, all the way up and down the tube. And then the longitudinal muscle is the one that is on the outer side, which runs lengthwise along the muscle. And together these are going to help propel food by contracting and increasing motility of the alimentary canal, and we'll see different ways that plays out here in a minute. Okay, and then the last lining is the serosa, and that's basically just a simple squamous epithelium embedded on top of a relar connective tissue sheath. Now, another interesting thing about the mesentery or the alimentary canal specifically in the small intestine is that there's this infolding of membrane that comes down and wraps around the tube and then comes back and anchors onto the dorsal part of the abdominal cavity. And that double infolding of lining is called the mesentery. Recall we had like the inferior mesenteric and the superior mesenteric arteries, and there's also the superior mesenteric vein. Well, those vessels, they run in that sandwich of membrane, and that's found anywhere along the small intestine, and then other portions as well, but it helps to serve as a place for blood vessels to get in and out, and then also keeps the guts or the viscera mostly localized in the same spot. Okay, now all the way up and down there are a few digestive processes that need to occur to make everything work. There is going to be propulsion, which is just simply the act of things moving along the tube. And the predominant way that this can happen is a movement known as peristalsis. Peristalsis looks something like this. The way that I think about it, it's like when you are going to squeeze a tube of toothpaste, and you squeeze the tube and then push it forward. That's an act of peristalsis. It's a wave-like contraction that sweeps along and propels something. And you see peristalsis not only in the digestive system, you see it in the reproductive system, you see it in the other places where there's ducts that have to move things in a short amount of time. You'll generally see peristalsis, is provided that there's muscle wrapping around the tube or the duct. So it's a pretty common way things are moved throughout the body. Okay, above and beyond just propelling stuff through, a major role of the digestive system, as you can imagine, is digestion. And this happens in two forms. One would be mechanical digestion. This is simply just breaking down the large bits into smaller bits. And a majority of the digestion that is going on here in the beginning portions of the digestive tract is mechanical. So you can see you have a lot of mechanical digestion happening in the stomach. One of the main jobs of the stomach is to twist and churn and churn and twist and mix everything up and break it down into smaller bits. From here then, there is an injection right about here, right past the stomach. There's an injection of a whole bunch of enzymes that are going to work to chemically digest the food. But even though most of the chemical digestion occurs here in the small intestine, it also occurs even as soon as the oral cavity. For example, you have salivary glands that are releasing saliva, and in that saliva there's an enzyme called salivary amylase, and that's going to help start digesting carbohydrates even as soon as the food touches your saliva. So chemical digestion really is occurring, as you can see, anywhere there's these little green things here. Is showing you most throughout the entire digestive system you find chemical digestion occurring. Well, once things have been broken down and once things have been chemically digested, oh, you know what? One of the things I was going to mention too, so chewing and churning of the stomach help to aid mechanical digestion, but also there's this type of contraction known as segmentation contractions, and those are shown here, which help to mix and churn things as well. And this is a little bit more of a complicated maneuver here. What we have is we have parts of the alimentary canal pinching and forming these little portions of pockets of food stuff, and then the pinch points release, and then they change their location. And so then, as you can imagine, if you pinched a little of this and pinched a little of this, you would get a mixture of the two boluses of chyme, is what it would be called. Chyme is the name that we give the solution of food stuff that's just dissolved and has passed out of the stomach. Okay, and then if this was to repeat, that these pinch points would relax and then they would pinch again, now you're starting to mix this all together. So it's like these alternating pinch points along the muscle there that compacts and churns things as well. Okay, now after everything's been mechanically and chemically digested, then the final goal is to absorb. And it's here where I like to just stop and appreciate the fact that literally, like, everything in you, everything that composes your body, everything that is inside of you, was absorbed through the digestive system. There isn't really another, apart from maybe oxygen coming in from your respiratory system, there really isn't another means by which you get building blocks to your body to sustain and build it. So absorption is a major important part here. Everything that is going to make up you at one point was absorbed from your digestive tract. And specifically, this would be absorbed across the epithelial tissue of the small intestine and the large intestine. Okay, the stomach looks something like this. You have the esophagus, and food will come down through the esophagus, and there's going to be a ring of muscle here, which is called the gastroesophageal sphincter. And that will act as like a gate into the stomach. And when the food bolus of blood passes down through the esophagus into the stomach, it will close off and prevent regurgitation. It will prevent things from moving backwards. There's a lot of acid in the stomach. The stomach is like a pH of 1, which few are the things in the world that are that acidic. So it's a very acidic environment. And closing off the gastroesophageal will help to prevent that acid from going backwards and scorching the esophagus. Now, this can sometimes happen, and then we call it acid reflux, or we call it heartburn because it sort of feels like the pain is coming from your heart, which in fact is the name of this region of the stomach. It's called the cardia because of that reason. That when there is pain experienced here, it's often referenced similarly so that it feels like it's coming from the heart. So here we have the cardia. There's a little bump on the stomach known as the fundus. And fundus is somewhat of a general word. You also see a fundus on the uterus, which is a similarly shaped portion here, like a little bump. Okay, there is the body of the stomach is the predominant part of the stomach. There is folds inside the lining of the stomach. And so what happens is that the stomach can stretch, and then when it retracts, there are all these wrinkles that form, which are called rugae. There is on the distal end of the stomach a part known as the pylorus, and the pylorus has the antrum, which will be like the narrowing into the final exit, and then there is going to be a really important ring of muscle, which is known as the pyloric sphincter. The pyloric sphincter closes, and it prevents things from getting out of the stomach prematurely. You can imagine that your stomach would not be very good if the food came in and then shortly thereafter it came right out. Okay, so there won't be any digestion that's occurring there. So being able to shut and seal off this outlet is a really important feature of the stomach. Okay, now if we looked at the lining of the stomach, it looks something like this. It's mostly flat, but there are these craters in the lining of the epithelium. There are these infoldings of the epithelial tissue which create gastric pits, and the gastric pits give way then to gastric glands. The glands are just a collection of cells of which there's some unique cells that can do special things. So the three types of cells that are in a gastric gland are parietal cells, chief cells, and enteroendocrine cells. Now, parietal cells will make hydrochloric acid, and this is done by the following mechanism. What happens is the parietal cell, so here we have a parietal cell, will take carbon dioxide and water, and through an enzyme that we should know by now, we saw this in the respiratory system, carbonic anhydrase, it will create carbonic acid. Then from carbonic acid, it will dissociate into bicarbonate ion and hydrogen ion. Well, a clever mechanism here that makes it such that you're not creating acid inside the parietal cell. Like if we were making a bunch of hydrochloric acid right here inside the cell, it'd probably scorch stuff. And generally you want to keep pH quite maintained inside of a cell. There's a lot of enzymes and proteins that are highly sensitive to pH, so changing the pH might denature them and render them ineffective. So the clever mechanism at play then is from carbonic acid, or sorry, the dissociation of it, the hydrogen ions are pumped straightway out into the lumen of the gastric glands. But the bicarbonate ion that results from this dissociation has a little bit of a greater journey. It gets pumped out of the cell with a chlorine antiporter. So the bicarbonate ion is exchanged for one chlorine. Okay, now the chlorine's got to diffuse all the way back, and it does so. It leaves the cell then passively, flowing down its concentration gradient. So this is just a chlorine channel. And now you have hydrogens and chlorine ions in the gastric glands, so they are together, working together to form hydrochloric acid. And it took somewhat of a delay for that to happen. Well, then the hydrochloric acid is secreted out through diffusion. It passes through into the lumen of the stomach, and there it has a special role, because what it does is it helps to activate the thing that's made by the chief cells. So the chief cells are going to be much of the cells that line the gastric gland, and their big product that they make is something known as pepsinogen. And pepsinogen is the inactive precursor, but when it is in the presence of hydrochloric acid, it often gets converted into the active form, which is known as pepsin. Okay, pepsin is a protease. It digests proteins. So we already started in terms of the digestive story. In the mouth, we've already started digesting carbohydrates with salivary amylase. And then the first place where we start to ramp up digestion is in the stomach. And the principal thing that we're digesting in the stomach is proteins. Proteins are more complicated. They're harder to digest than carbohydrates. So there's an extra bit of effort given here in the stomach to digest them. There's also an important mechanism here. It's an important thing that hydrochloric acid is required to convert pepsinogen to pepsin, because it creates sort of a limiting environment to govern how this is working. Proteases are a fickle thing. You don't really want proteases to be created in large abundance in your body, because they basically start to destroy your body, right? There's a lot of protein in your body. There's a lot of protein in your cells and in your cell membranes, and if you start eating that away with proteases, your cells are going to turn to mush. So one of the things that's helpful here is that pepsin works in a very high acidity. It works in conditions of low pH, and then it stops working once the pH is altered. And so this is helpful, because when all of that chyme leaves the stomach, you don't want that pepsin probably still working anymore, because now it's going to start to erode away all of the cells lining the small intestine and all the cells lining the large intestine. So the body very quickly neutralizes acid distal to the stomach, and thereby inactivates pepsin. Okay, but in the stomach, it's a great thing. Lots of acid, lots of pepsin being made to make lots of proteins. There are also enteroendocrine cells, and these cells, as the name suggests, enteroendocrine means a cell that is part of the endocrine system that is found along the digestive tract. These make there's a variety of hormones these can make, but one of the big ones is, specifically in the stomach, one of the big ones is this hormone known as gastrin. Gastrin is like the big get stuff going in the digestive system hormone. And when these cells are stretched because food has just entered the stomach, or if these cells are given a stimulus from the vagus nerve, let's say, they will release gastrin, and that will cause the muscles of the muscularis externa to start contracting and churning. The stomach will also be a signal for the parietal cells to start secreting acid. Now, a quick clinical application here for the parietal cells is acid reflux. So this is when you have an elevated production of acid, which erodes away the mucosa of the esophagus. Generally, the esophagus, stratified squamous epithelium, so here's the tube. It's going to be coated with a mucus like this, and a mucus like this, okay? And that mucus is going to protect the epithelial cells from any acid which might regurgitate from the stomach. We call it a mucosa. Mucosa just means epithelium plus mucus. So the mucus is on there, so we're calling it a mucosa. Well, if there's chronic elevation in acid produced, it's going to erode away that mucus, and then it might actually start to eat away at the cells lining the esophagus, and then maybe start to scorch some of the nerve receptors or the submucosal plexus, and that can cause a lot of pain. Well, one of the workarounds, this is the parietal cell again. one of the workarounds to prevent acid from being made, or to limit how much acid is being made in the gastric gland, is to shut off this pump. This pump is called a proton pump. And so consequently then, any agent which turns off that pump is called a proton pump inhibitor. And there's several classes of these, like omeprazole and I'm trying to think of some others. Well, one of the tricky things about this, though, is that acid is required to have pepsinogen converted to pepsin, and that so it's required in the protein digestion. So if you start limiting the acid that's produced, then it can start to limit how much of the proteins are digested, and then that can start to skew the efficiency of the digestive system. Well, this is tricky then, because people will use a proton pump inhibitor to reduce the acid, and it will make them feel better. They won't have that sharp pain of the acid scorching nerves and things in their esophagus. But if they start to experience problems with digestion that result from the decreased efficiency of pepsin, then they might say, you know what, I'm done with the proton pump inhibitor. I'm not digesting my food as I ought. I'm feeling it. I'm feeling these side effects. So they'll go off the proton pump inhibitor. Well, if your cell has been inhibiting these proton pumps, likely the cell is responding by upregulating the expression, meaning inserting more of those pumps in because they're not working, because they're being blocked. So now, if you do away with the proton pump inhibitor, you may have an upregulation of that proton pump, and now they're all unregulated. They're going to start pushing acid out like crazy, and that will cause a severe amount of pain. So even though proton pump inhibitors are not addictive, they sort of can be, because if you get used to using one and you go off of it, it can be like a gateway, a threshold of immense pain that awaits on the other side of going off of the proton pump inhibitor. So I think generally it's really important before going on a proton pump inhibitor to try all the other solutions to treat acid reflux. And acid reflux, heartburn, can be an excruciatingly painful thing. One of my favorite remedies is good old baking soda. So whenever I have an upset stomach or I feel sick, I always put a teaspoon of baking soda in a glass of water and chug it. And what you're doing is you're putting a bunch of this into the system, and you're putting a bunch of bicarbonate ion in, and when that bicarbonate ion reaches your stomach, it attaches to all of the hydrogens, which then form carbonic acid, and then that dissociates to form water and CO2. It's just the reverse of what we're seeing here. Okay, so putting bicarbonate ion in will drive the reaction the other way, and now you're creating water from the acid, and CO2 is a byproduct. And you'll notice if you do this, you'll have like these big burps that feel like you just drank a bunch of soda. You have a bunch of carbon dioxide you have to get rid of. So that is my solution, and I would encourage that to be something that people try, or like Tums and things like that. Those are also forms of bicarbonate ion, but I think the good old-fashioned baking soda down the hatch is the best. Fair warning, if you try it, it tastes like a dead fish. It is horrible. I don't know if you've ever drank baking soda water before. It is horrible. So you just got to chug it down as quick as you can. All right, all interesting stuff here concerning acid reflux. Okay, another thing that parietal cells will make is intrinsic factor. Intrinsic factor helps you absorb an important vitamin known as B12. And B12 has a variety of roles, but it's generally well known for being something that helps you to create the myelin sheath around your neurons, as well as it plays an important role as a cofactor in the maturation of red blood cells. For example, like pernicious anemia, a type of anemia that is going to result from a deficiency in people that are deficient in B12. In either case, there was the chief cells, we said pepsinogen, and the enteroendocrine cells produce gastrin. Okay, the phases of gastrin secretion. How is gastrin secretion regulated? What is it that turns it on and off? It starts with the cephalic phase, and cephala is the root word here, which means head. So this is everything to do with your brain. We haven't even gotten anything in the stomach yet, and yet we can still prepare the stomach for some activity. So this begins with your brain. If you see something that looks pleasing, or the thought of food, or you smell it, or you have maybe even a small taste of it, these different perceived stimuli will ultimately land on the hypothalamus and the medulla oblongata, and via the vagus nerve will cause the stomach to start making gastrin. And when gastrin is made, things like acid are produced and the stomach starts to churn. On the flip side, to turn off gastrin secretion, if you had loss of appetite, which was, second to depression or other conditions, then you would not be releasing as much gastrin. So just what's happening in your brain and in your thoughts can modify the level of gastrin secretion. Okay, there is the gastric phase, and this is involving the stomach. So what further increases gastrin secretion would be if the stomach is distended. That will activate the stretch receptors. Other signals would be food chemicals and rising pH levels. There's chemoreceptors that are inside your stomach, and there's chemoreceptors that are all along the alimentary canal, and they're monitoring pH. And so when stuff enters your stomach, the pH rises because the acid's being diluted. And so either of those chemical signals or those mechanical signals, ultimately through different pathways, will do the same thing. They will cause gastrin to be released and the stomach to start churning and releasing more and more acid. Okay, then there's the intestinal phase, and so this is how gastrin secretion is usually the feedback pathway, how it's shut off, but it can also turn it on. But it all has to do with what the situation is in the duodenum, what's going on in the intestines. So for example, if there is the presence of a low pH or partially digested foods, then gastrin is released in the blood. That makes its way all the way back to the stomach, and more activity is encouraged. If there's a distension of the duodenum, meaning a bunch of chyme has just came out in a bolus, which would generally happen if there was sufficient digestion and churning of the contents in the stomach, well, then this would shut the whole thing off and inhibit the stomach activity. There's a great table in the book which summarizes all the different hormones that the stomach makes, and thus far we've talked mostly about gastrin. So here we have gastrin. It's made in the G cells, predominantly in the pyloric antrum, so that's that distal portion of the stomach. Here are the reasons why it would be made. If there's peptides or amino acids that are in the stomach, that would suggest that there's something, that food is in there and it needs to be digested. Now, gastrin can have different targets, though, and then based upon those targets, slightly different roles. So if the gastrin makes its way back to the stomach, this will increase the secretion of the gastric glands and promote gastric emptying. If it reaches the small intestine, it will promote intestinal muscle contraction. If it reaches the ileocecal valve, it will release the valve. And if it hits the large intestine, it triggers mass movements. All of these are encouraging movement of the chyme through the alimentary canal. We'll see where these, like the ileocecal valve and stuff, we'll see where that is. They're all checkpoints and gateways that encourages food to move through. Okay. Another hormone is ghrelin, and ghrelin is made in the fundus, and it will stimulate hunger. So it's thought to be like the hungry hormone. Okay, there are some others here, like histamine stimulates the parietal cells to make hydrochloric acid. Serotonin, also made in the stomach, causes contraction of the stomach muscle. Somatostatin, made in the stomach, different regions, restricts restricts the gastric secretions, so this would be like an antagonistic hormone. And depending on where it's going, it can have some different roles. Okay. Generally, though, what I want you to know from this table is what ghrelin does, and then also what gastrin does. And generally speaking, gastrin increases the motility of the food that's being digested. One last thing to mention here before we move on, there's a class of what are known as bariatric surgeries, and these would be surgeries that are aimed at changing the stomach to encourage weight loss. And these are surgeries that they're not uncommon surgeries. I know a few people that have had these surgeries done. There's different forms of them. On the one hand, what they could do is make an incision along the stomach and basically just shrink the stomach down so that it can't expand as much. And in so doing, a lot of the portion here of the fundus, the place where that ghrelin is released, is excised and removed. So less ghrelin is made, there will be less hunger, and then there's just less room for the food to go in, so you're hungry less, you feel fuller more quickly, and that, as you can imagine, would be a really significant way to decrease your your food consumption and your weight gain. There's also what's known as a lap band, and this is a band that wraps around the stomach, and the diameter can change, and there's actually a little port that you can inject hormones in to constrict or relax that band, so it opens and closes the gateway into the stomach. And then there is the gastric bypass, and in this surgery, what they do is they take a portion of the stomach, just the very proximal portion, the cardia, and they'll take the intestines and they'll just pull them right up and bypass the stomach altogether, and then suture them together and cauterize them together. And then the stomach then gets cauterized, so the stomach is still intact. It can still make things like gastrin, and those can still make their way down into the intestines to help absorb, but a lot of the storage site here of the stomach has been reduced. The food just goes straight down into the stomach. And these surgeries have varying levels of, like, some of them are more reversible than others, and some of them are more severe than others, but at any rate, a variety of different surgeries that could be done, all aimed at weight loss. Okay, moving right along then to the small intestine. The small intestine's got three parts to it. There's the duodenum, there is the jejunum, and there is the ileum. The duodenum is just this first part coming off of the stomach. It's only about 12 inches or so, just this first little C-shaped part. The duodenum is not very well suited for absorbing nutrients. The small intestine as a whole is the major site for absorption, but not really the duodenum. The main job of the duodenum is to neutralize the acidity of the stomach. There's lots of mucus that's shrouding the tissue here and protecting it, keeping it from getting scorched by that acid. And because of all the mucus, it's not really all that great at absorbing. So if the acid can be neutralized here, then in subsequent portions of the small intestine, there doesn't need to be as much mucus, and there can be more effort undertaken to absorb things. And that's what we principally see here in the jejunum and the ileum. Now, all across the small intestine, the name of the game, is specifically in the jejunum and the ileum, is to increase surface area. Increased surface area lends for greater absorption. And there are three features that allow that surface area to be greatly extended above and beyond what normally would be. There's a statistic out there that the total surface area of the intestine, if you unfurled it, would be about 250 square meters, which is about the size of a tennis court. In contrast, if you just took the small intestine as a tube without all of the additional surface area, it was just a plain old tube, I mean, that surface area would only be maybe a couple square feet. So having all the folds in the villi and microvilli greatly enhance how much area there is for absorption to occur. Let's take a look at what these features appear as. So on the left here, we're seeing the alimentary canal. We're seeing a cross section of it, and here we can see the circular folds. So there's a folding of the tissue inward, and that wraps all the way around, and it forms these little ribs or folds all the way around. And that would be seen here. Here's like the histology showing that. You're seeing this would be probably the submucosa here, and this would be the or maybe the myenteric. No, this should be the submucosa. And the mucosa is coming in like this, forming these circular folds. Well, on the individual circular folds, so this would be like an extension of the tissue forming the circular fold, there are additional little extensions off of that, which are called villi. And that's what you're seeing illustrated here. So if you were to take a little snapshot of one of these little villi and magnify it, this is the illustration here. Now, in each villus, there are these extensions of lymphatic vessels called lacteals. And the lacteals will be one of the places that lipids are absorbed. For whatever reason, the lipids aren't once they get across the cell and into this space just behind the epithelium, they're not absorbed very well into the capillaries of the blood. They instead go into the lacteals, travel in the lymph for a while until they rejoin the blood in the ducts that enter just by the internal jugular vein on either side of the body. Okay, there's also going to be a lot of capillaries waiting behind here. Again, this layer of cells, the epithelial layer of cells here, is the boundary through which things must pass to become part of your body. Anything that's out here is not part of your body. As soon as it crosses the cell and the cells permit it to do so, now it is part of your body and can be incorporated into building it or reconstructing it or whatever. So I always like to appreciate that you've got one cell between that which is you and that which is not you. I think that's pretty cool. All right, and occasional places you'll see these little pits. They're analogous or similar to the gastric pits. These are called the intestinal crypts. And in these crypts, there's a variety of cell populations that can be found. Some would absorb salts and things. Some might be goblet cells releasing mucus. Others might be enteroendocrine cells creating hormones. So it can vary. But they would be found in these crypts. Then, if we looked at the final feature that encourages greater surface area, on each individual epithelial cell, so we're talking about one single cell here, we blow that up. If you look at the plasma membrane, it has a similar appearance as the villi. The plasma membrane itself is undulated or waving, moving up and down to create the greater surface area. We call this feature the brush border because when you look at it under a microscope, it looks almost like there's looks like a little brush. We also call it that because what happens is that on the apical side of these cells, embedded in this membrane of the microvilli, are a variety of receptors that can absorb nutrients, and then additionally, there are some enzymes that are made here, which are called the brush border enzymes. We'll look at those later. But the brush border enzymes generally are the things that are going to do the final leg of digestion. So let's say you had a big, long carbohydrate, maybe hundreds and hundreds of glucose molecules bound together. By the time it gets to here, that big polysaccharide with all of the glucose molecules would maybe be just small oligosaccharides, maybe 3 or 4 linked together, maybe like up to 10. Well, the brush border enzymes, they're designed to just cleave off the small, the final bonds between the monomers, and ultimately, then what can be absorbed across this membrane, the thing that makes something you, is generally only monomers. So think about that for a second. This big, complicated mixture of chemicals that is in something like food. Broken down. By the time you get to the brush border, only single individual monomers of those complex assortments of proteins, lipids, and fats, and nucleic acids, only the monomers are absorbed. And generally, like even a disaccharide might not be able to be absorbed. Even that has to be broken down. So it's only single little monosaccharides having their own little receptors that enter into the cell. So that's the final place that digestion happens there, along with brush border. And we'll look specifically at what some of those brush border enzymes are more in the physiology portion, which is later to come. All right. Now, here's where we have to pause and talk about some accessory organs of digestion, because in the small intestine, in the duodenum, there is the pancreas and the liver that are going to create important things and put it into the duodenum. We'll talk about the liver first and work our way down through. Okay, so the liver, what's going on here is the liver is arranged in what are called lobules, and a lobule looks something like this. So imagine this structure lengthwise going up and down and packing a whole bunch of those together. That would be what your liver tissue looks like, the microanatomy. And a liver lobule is going to be sort of a hexagonal shape, 6 sides, and at each point there is what's known as a portal triad, which is going to be 3 different vessels. The 3 vessels that are found in the triad are the following. You have a branch of the hepatic portal vein. So just remember more macroscopically here. Remember you had the liver, and you had the splenic vein. You had the mesenteric vein, superior mesenteric vein, and you had some other veins as well, smaller ones. But in essence, all of the blood that's collected from the viscera gets gathered into this one vessel, which is known as the hepatic portal vein. Okay. Well, what happens to that vessel is it goes into your liver, it branches, it branches, it branches, and branches, and then it has all these little small branches that find their way circumferentially around the hepatic lobule. So that's what these blue ones are that you see here. They're branches from the hepatic portal vein, so we call them portal venules. Okay, on the flip side, you also had, this is sort of an interesting thing that you don't see often. Remember when you had the aorta and you had the diaphragm, and one of the first branches off of the aorta was this trunk that had 3 branches off of it. That was called the celiac trunk. And the 3 branches off of it were the gastric, the splenic artery, the left gastric artery, and then this one was the common hepatic artery. Well, the common hepatic artery branches into right and left, and then branches again, and eventually those branches end up here as the portal arterioles. So the portal arteriole is a branch off of the common hepatic artery, off of the celiac trunk of the aorta. So it's an interesting thing that happens because you have, on the one hand, portal blood that's just been from the guts and now is nutrient rich, but oxygen poor because it's already been to, specifically here, it's been to these capillaries and it's lost the oxygen into the tissues of the mucosa. Okay, and so by the time it reaches the liver, that portal blood is oxygen poor. Lucky for you, though, there is a little branch that runs right next to it that carries oxygen-rich blood, and together these dump into the same network of capillaries, which are sinusoidal capillaries. Remember, we said sinusoidal capillaries are the most permeable. They will have larger gaps and cracks in the lining of them, so that bigger products like plasma proteins and things can get into and out of the sinusoid. Well, that's what we're seeing here. This network is going to carry partially oxygen-rich blood because it's got some oxygen-rich blood and some oxygen-poor blood. And that all roads of those, all of that network of sinusoids all gets brought into what's known as the central vein. And then from here, all the central veins collect, and macroscopically again, the central veins of all these lobules connect and then they pool into a variety of veins, which are going to all dump that blood into the inferior vena cava. And these individual vessels here are called the hepatics. They're the veins that drain all of that blood from the liver that's collected through the central veins. So an interesting vascularization here in this organ. Now, there's also then the third thing in a portal triad is the bile duct. And the bile ducts have these ductules, they're called the bile ductules, which extend into the hepatocytes. The hepatocytes are all the cells that are living here. And wrapping around every cell, there's like a little moat, you can think of it, that collects bile produced by these cells. And that bile will get brought down through the ductules into the bile ducts. And then from here, you've got the liver, and you've got, sorry, number 3 of a pathetic liver drawing from Professor Stelter. Okay, well, here we go. All of the bile is collected from all of these bile ducts coming together. And then it all gets brought into what's known as the common hepatic duct. From here, it continues on into forming what's known as the common bile duct. But there's a little offshoot, there's a little branch here off the side where there's an organ called the Gall Bladder. And that branch is called the cystic duct. Okay? And then from here, this gets brought down into the duodenum. All of that bile will come down. So lots of tubing, and I always tell students that in another life I'd probably want to be a plumber, because when I see all this, like I love fluid flowing through tubes. I think it's cool. If you come to my house ever someday, you'll see my radiant floor heating system, 1,500 linear feet full of fluid that heats my entire home. My home literally has a circulatory system to it. I just love fluid running through tubes, and when I see this is a lot of tubes with a lot of fluid running through it. And to think that all this is arranged similarly for every lobule. It's not just random sporadic vessels. They are highly organized units packaged together. And all of that from one cell. You know, you've got one cell making this whole thing, and all that information prepackaged into that cell. I mean, that is nothing short of a miracle. Okay, so back to some stats here on the liver. It's composed mostly of hepatocytes. That would be these cells here that make bile. They also make a variety of plasma proteins like albumin, one of the main proteins that establishes the blood colloid oncotic pressure. That's made by these cells and then transported into the sinusoidal capillaries. They will process the nutrients for you, like they'll take the glucose monomers out of your blood and put them back together again, reassemble them to form glycogen. A lot of glycogen is made in the liver and stored in the liver. They will detoxify things. If you look at the smooth endoplasmic reticulum in these hepatocytes, that's quite large. That's an organ that's well suited for detoxification. A lot of your alcohol gets detoxified here, as well as any other drug, like even like acetaminophen or Tylenol. Generally, it will end up in the hepatocytes to be processed. And different waste materials will be processed here as well. Like I think there's something to do with ammonia, I think, that gets processed in the liver. Okay, this is also a place where proteins and lipids and carbohydrates are stored. And then we have the Kupffer cells. Special note here about the Kupffer cells. The Kupffer cells are phagocytes that unfortunately are not found in this drawing, but they would be phagocytes that are found along in these sinusoids, and they're there waiting to remove any red blood cells or white blood cells that might be damaged. Okay, one of the things that also happens in the liver is that to make bile, what the precursor molecule that the hepatocytes are going to use to make bile is something that's known as bilirubin. And here's a review question for you. Where did the bilirubin come from? Bilirubin comes from something that's known as biliverdin. And biliverdin comes from the non-ferrous heme groups of hemoglobin. So you take hemoglobin, it's got 4 heme groups. Each heme group's got an iron atom on it. Take off the iron, and then you're left with the heme. The heme gets converted into biliverdin. Biliverdin gets converted into bilirubin. And this is the thing that can accumulate in your skin to create jaundice. And then it's also the name of one of my friends in high school. I don't know if his parents knew about that when they named him bilirubin, but that then gets converted into forming bile. Okay, a couple of clinical applications here just for interest's sake. A colic stool would be a stool that is not brown in color. So normally what happens if there's bile, which there should be, the bile gets converted into stercobilin, and this is done generally by bacteria, I think. And stercobilin is brown. And so this is what makes your fecal matter brown. It's ultimately the bile that's made from your heme groups of your red blood cells. Well, if there's no bile, then if you have a stool with a higher fat content, it can appear to be lighter in color. And fortunately, I don't have any visuals for you here. But yeah, normally your feces should be brown, and if it's not, if it's white or pale light, that's probably something to do with the amount of bile that's being produced. Okay, the gallbladder, we'll take a quick look at this. We said that the bile that's collected from the hepatocytes comes down, it gets stored in the gallbladder. The gallbladder is something of a muscular organ. It actually has muscle in here that can contract under the influence of certain hormones, which we'll talk about in a moment, and inject that bile into the duodenum. By the way, I guess I should say what bile does. Bile, what it does is it's an emulsifier. And so it takes a larger droplet of fat or oil, let's say, and it breaks it down into littler droplets like this. It does this because bile is amphiphilic, meaning there's a portion of it that's a little crystal, there's a portion of it that is hydrophobic, and there's a portion of it that's hydrophilic, and so it can bind to the oil, and then the water can carry it away and create smaller droplets. Okay, this is going to be the target site for an enzyme or a hormone known as cholecystokinin, or CCK. And so when it binds here, it contracts the muscles of the gallbladder, and that can inject the bile down. A clinical application here is cholelithiasis, and this would be gallstones. So for whatever reason, here's a jar of them. I don't know why you'd keep a jar of these on your shelf, but for whatever reason, there can form these little stones that are collections of like bile crystals aggregating together to forming a larger stone-like substance, and that would cause a lot of pain. And generally, if somebody has gallstones, they have them in their gallbladder, they can go in and actually just remove the gallbladder altogether, because the gallbladder is just a storage site. And so if it's removed, bile will still flow down into the duodenum, but it just won't have as well adapted of a regulatory mechanism here to store it into the gallbladder. Okay, on to the pancreas. One of the things to note about the pancreas is that most of it is exocrine. Okay, so there are these collections of berry-like features here that have cells in them. These are called the acinar cells. Acinar means berry. And they will be creating a whole host of things that then get collected into a duct. Well, because it's being collected into a duct and eventually making its way into the small intestine through this little port, it's called exocrine. We generally don't think of the pancreas being an exocrine organ. You generally think of it being an endocrine organ. And it is an endocrine organ, but a very small fraction of the cells in the pancreas are actually endocrine. So this is a cool illustration here. It shows you the difference. Right on this side here, we have what's known as the pancreatic islet, which means island. And you have a couple of powerhouse cells here that are surrounded next to a blood vessel, and they're going to make hormones, and those hormones go into the blood vessel. Some of the cells that you'll see here are the alpha cells, And then there's also the beta cells. The beta cells will be making a hormone known as insulin, and the alpha cells make glucagon. And they do different things. Insulin will pull sugar out of the blood. Glucagon will pull it out of your cells and put it back into the blood. So between the two of them, they can regulate your blood sugar. So those are made there. But here, this is all exocrine stuff, and so there's a whole variety of enzymes made here, which we'll talk about in greater detail later. There's also bicarbonate ion is made here in high abundance. Bicarbonate ion is going to absorb all the hydrogen ions coming out from the stomach. So remember, this is the duodenum here. Lots of acidity coming off the stomach, and we want to neutralize that. There's a steady stream of bicarbonate ion being injected right through this port into the duodenum to neutralize that acid. It's a very important step in the whole process. Otherwise, we're going to scorch the epithelial tissue further down, and moreover, there will be active pepsin in that chyme eating away at the proteins in the epithelial tissue if we don't neutralize the acidity. By the way, this is sometimes called the hepatobiliary tree because it's a branch point where the liver and the pancreas come together. And so there's this sphincter here, which is called the I better write this down. The hepatopancreatic sphincter. And it also has, they generally are straying away from these old school names, which are named from doctors from like the 1900s and 1800s that discovered these things. But this is one I think we should keep around. This sphincter is also called the sphincter of Oddi. And I think this is a great name. So you have full license to use that name as the name if you so desire. But the hepatopancreatic sphincter, the sphincter of Oddi, they both are going to govern contents from the liver and from the pancreas coming into the duodenum. So they can close to prevent things from passing into the duodenum, or they can open to allow things to pass through. You will make about 1 liter per day of pancreatic juices in your pancreas, and in that is going to be sodium bicarbonate that reduces the acidity. It also inactivates pepsin. One of the other enzymes is trypsin. This is similar to pepsin. It's a protease. But it's a little bit of a weaker protease, and it's more well suited for neutral pHs. So not quite as a heavy hitter as pepsin. Okay, amylase is the one that digests carbohydrates, and then you will also have a pancreatic lipase that is made, and this would be to digest fats. So the story with fats, we'll talk about it again here, but it gets broken down into little droplets. The fat does, or the oil. And then from here you have the lipase coming and breaking apart the fat molecules into individual little components. Okay, there are also nucleases here which will break down DNA and RNA. So a whole assortment of enzymes made here to break down really every class of molecule that exists. Okay, I think we'll stop here at the large intestine and we'll pick it up again on the next part.